All right, we're going to give it about 30 more seconds, guys, then we'll get started. How is Dead Week Eve going for you guys? Good and stressful. I hear that. Excellent. All right, guys. Well, we are not going to waste any time this morning because I want to leave some time to field some questions about group projects. No poll everywhere stuff this morning. So we're going to go ahead and jump right in. I'm going to share my screen with you. We're going to look at Blackboard, and then we're going to switch over and just briefly run through an abbreviated lecture from chapters 12 and 13, which is what we're focusing on this week. All right, so with Blackboard, there's a couple things going on here. So first and foremost, we are in week 13. Nope, week 14, that's right. And so this week is kind of our last big push of new material. So next week will just be a really abbreviated lecture about parturition. And so this week we are going to be talking about sperm cells journey in the female tract after copulation. And then we're going to be talking about fertilization as well. And so, fertilization is going to be part of that sperm in the female tract lecture. And then we'll talk about early embryogenesis and maternal recognition of pregnancy. So early embryogenesis is going to be something that is actually familiar to you guys because we talked a lot about early embryogenesis in chapter 4. So that'll be a good refresher for you. And then maternal recognition of pregnancy will be new. And so you have 2 quizzes to complete this week. So chapter 12 and chapter 13. So just make sure that those are on your to-do list. And then the other big thing that is due this week, which we've been talking about for the last 10 weeks, 11 weeks, is the semester project. And so I know I have talked to a lot of groups, a lot of individuals that have come for mentoring and asking additional questions and defining goals and strategies and all of that. And so I very much appreciate those of you guys who have made the effort to make an appointment for office hours and talk through some of those. And so in the semester project folder over here to the left-hand side underneath start here is where, of course, you'll find the assignment. So I'm hoping you've already seen that. And then there's some example business plans from the ANSC 471 class that are similar to what I am expecting. But keep in mind, what I am fully expecting is the rubric that is in the actual assignment. And then you will also be submitting the semester project through the link here in this folder. So the link to the project submission is in the semester project folder, not in the week 14 folder. So please just keep that in mind. Also, when it comes to submitting the project, please keep in mind that everybody needs to submit a copy of the project. So that's really directed to those of you guys that are in groups. So yes, you may be completing the assignment as a group, but that means that every group member turns in a completed copy of that assignment. And so the reason for this is because I want to make sure that everybody ends up having a grade for that semester project. In the past, it's been really convoluted. and really complicated in the online format. I could handle it better when we were in person and you guys were handing me like a stack of papers as a plan. But with the electronic version, it just gets really complicated really quickly. So please, everybody needs to turn in a copy of the semester project. If you don't turn in a copy of the semester project by the due date and time, the late policy will still apply, right? So if you forget because you think somebody else is turning that in for you and you forget to turn and you don't get it turned in until like Saturday night or Sunday night, then the late policy percentages will still apply. So just please keep that in mind. So with that, that's just an update on where we're at from a Blackboard and class perspective. Can I field any general questions about timeline, how the semester is wrapping up, or anything like that? I'll give us just a minute here to share some of those questions. Would love to talk to you, or you can, of course, put them in the chat. All right, sounds good. So we're going to go ahead and transition and talk about chapter 12 material. So let me get to that file. Let me make sure I'm actually sharing the right screen. So that's the one we need. There we go. All right, so chapter 11 last week was all about reproductive behavior, and you guys completed that case study. And so chapter 12 really takes us into what that behavior is attracting in the events that are going on inside the female during that point in time. And so with this information, what we're really looking at is what happens to the sperm cells that are deposited into the female's tract after that copulation event. And so from a general overview perspective, what's actually happening is once the sperm cells have been deposited in the female's tract, it goes into the first stage of sperm transport, which is immediate transport. So here we can have sperm cells actually leaving the tract. So they're swimming the wrong way. They get flushed out because of the contractions, all of that type of thing. Also in this area, we can see lots of phagocytosis happening, which is the cell death experience. So the sperm cells may be damaged. They may be old and have reached the end of their lifetime. And so they're going through the phagocytosis experience. Or we have sperm cells that are actually going to proceed into the entrance of the cervix or the uterus, depending on the species that we're talking about. And so with the immediate transport, that is occurring directly after deposition in the female's tract. Once the sperm cells are actually in the cervix, again, dependent on species, the cervix is a really complicated navigation system. So there's lots of muscle and structures in the cervix that help aid in keeping the fetus in the uterus during pregnancy and parturition. It also helps keep bacteria out because of this complex navigation path, but it can also be a hindrance to the sperm cells. And so we call it the privileged pathways. So if sperm cells are lucky enough to be able to navigate through the complexity of the cervix, then they are choosing that privileged pathway. And so the cervix is a really key player in this event because it can help remove sperm cells that are not motile and it can also help remove sperm cells that have abnormalities as well. So once the sperm cells navigate through the cervix, they are then in the uterus. And during their time in the uterus is where the process of capacitation occurs. We're going to talk more in depth about what capacitation is, but just keep in mind that happens in the uterus. We will also see phagocytosis continue to happen in the uterus as well. So keep in mind, there's quite a bit of time associated with the sperm cells actually navigating from their drop-off point to the point in the uterus. So that's, again, a timeline where those sperm cells can actually be go through their life cycle and die during that process. The fourth major stop along their journey is the oviduct. And so once these sperm cells are navigating through the uterine horn into the oviduct, specifically, they'll go through this uterotubule junction. Then that process of capacitation that was initiated in the uterus is completed in the oviduct. And what ends up happening here is the sperm cells actually become super hyperactive, like on caffeine. So they become really, really hyperactive and motile. And that's key for the fertilization process, which is the next step in this whole overview process. And so during the fertilization process, we see the acrosome reaction occur, and we'll talk more in depth about that. It, like capacitation, is a really important event in this pre-fertilization process. We'll also see the spermatozoon penetrate the oocyte at this point. That's the beginning of that fertilization process. And the kind of the crux or the climax point of fertilization is where the male and the female pronuclei actually form together and form that zygote. So that's just an overview of what is occurring to sperm when it's in the female tract. Keep in mind that there's a lot of loss of sperm cells along this pathway. So the biggest loss is going to be due to phagocytosis, and that's primarily going to be done through the neutrophils. So neutrophils are part of the immune system, and keep in mind, sperm cells are actually a foreign entity in the female tract. So the immune system is going to attack them at a really high rate. And so that's why concentration of sperm cells and the amount of total sperm cells getting into the tract becomes really important, because it becomes a power in numbers type situation. And then the second major loss that we see in sperm transport is from retrograde transport. So that means sperm cells that are in the wrong direction, swim in the wrong way, or just get flushed out of the female tract because of the contractions. So keep in mind that the female is very much in a standing heat at this point in time. So if we're talking about a natural state, then that female is going to be in a standing heat. The follicle is going to be the dominant structure on the ovary. It is going to be cranking out a ton of estradiol. Estradiol is going to be feeding back to the hypothalamus and the cervix and the uterus. And keep in mind that with the uterus, what it's actually doing is actually contracting and releasing those smooth muscle in the endometrial layer, or the myometrium of the uterus. All right, so just a couple more comments about the way that sperm are transported in the female system. It really happens in two major stages. The first stage is what we call rapid phase. So the sperm cells actually, these are going to be the sperm cells that arrive within minutes to the oviduct after deposition in the female tract. So that can happen really, really quickly. Typically, those sperm cells are not super viable. So they've been beat up a lot. They could be older. They could be damaged by the muscularis contraction. And so keep in mind that the sperm cells that show up at the in the oviduct at this point in time is still being beat around by high amounts of estradiol in the system. And so these are kind of the first responders. And so they tend to not be super fertile. In addition to that, we can also see that there are some prostaglandin proteins in the semen that will cause increased tone and motility of the uterus as well. So it's kind of a really unique thing that sperm can actually contribute to that contractile action. It's not just on the part of the female and estradiol binding to its receptors in the uterus and causing those contractions. The prostaglandins on the sperm cells or the prostaglandins in the seminal fluid can also cause that response as well. So that's one way sperm can actually show up in the oviduct. The second way is through the sustained release phase. And so this is where we actually see a trickle-like phase of sperm cells entering at lesser urgency to the uterus, to the uterine horn, and to the oviduct. And the sperm cells that are in that sustained release phase are really, really important for fertilization. Just a quick note on the mucus that is going to be secreted by the cervix. So there's two different types of mucus that will be secreted depending on where the female is at in the estrus cycle. So if the female is in estrus, meaning that there's a dominant follicle on the ovary that is cranking out a lot of estradiol, estradiol is going to attach to its receptor in the cervical cells and produce ciliamucin. And so that is this first type of cervical mucus. So that is going to be produced to aid in sperm transport. And so this helps create that slip and slide effect so that the sperm cells can navigate through those crypts in the cervix. So this is creating a sperm highway. The second type of mucus produced by the cervix is when the CL will be dominant on the ovary and will be producing progesterone. So the female will not be in a standing heat or an estrus at that point in time. Progesterone will be the primary steroid in the highest concentration in the blood system at that point in time and will bind to its receptors in the cervical tissue and produce sulfomucin. And so the sulfomucin is really important because it actually will layer all the cervical crypts in the cervix with a very thick viscotic mucus. And so this will actually help to remove sperm cells during this point in time. So it will catch sperm cells and it's going to be very, very thick during that time. All right. The next major event that occurs is capacitation. So capacitation is going to begin in the uterus and is going to end in the oviduct. So during this capacitation process, the sperm cell is going to pick up seminal plasma proteins. So these are going to be proteins that are part of the seminal plasma coming out of the male, but they aren't going to be picked up until the capacitation event. And so these proteins are what we call heparin binding proteins. And so these HBPs are going to bind heparin. Heparin is going to be present in the female tract. And once that binding takes place, those proteins are going to be pulled off the sperm cell, the head of the sperm cell. And this actually pulling off process causes the sperm cells to go into a hyperactive state. And so we have to have the sperm in place and seminal plasma that is coated in heparin binding protein for this to happen. And so in the reproductive tract, when HBP binds heparin in the uterus, it actually will remove seminal plasma proteins that were coating the head of the sperm cell, and will also expose the zona pellucida binding sites. And so the zona pellucida binding sites are going to be on the head of the sperm cell, and they are going to be kind of a docking station that will meet up with the zona pellucida of the oocyte. So that is coming in the next part of this process. And so that heparin binding protein action. So here's our proteins, and then this is them binding with heparin. And once they're bound with heparin, the whole protein will come off of the sperm head here. And we end up creating a sperm cell that is very, very hyperactive. And so that will then launch that sperm cell further into the oviduct, specifically to the ampulla isthmus junction of the oviduct, where the oocyte will be waiting for it to fuse together. And so that fusion process is the process of fertilization. So fertilization by itself is not an on-off process. It is not an on-off switch. It doesn't happen automatically. Fertilization is a process with multiple steps, and those steps are outlined over here on the right-hand side of this slide. So first of all, we have hyperactive motility because of the capacitation process. We'll see binding of the sperm cell to the zona pellucida. We'll then see the acrosome reaction occur, which we'll talk about more in depth in here in just a second. And then that sperm cell will penetrate the zona pellucida of the oocyte. The sperm and oocyte membranes will fuse at that point in time. The sperm will be engulfed into the oocytes, and then we will see a decondensing of the DNA occur in the sperm nucleus. And then finally, we will see the formation of the male pronuclei. And so that will result in the creation of a zygote. All right, so let me see if this animation will work. I'm hoping that it does. Okay. What is happening? Maybe it's not going to work. Okay, I will go back and I'll paste that animation in as just a link, since it doesn't want to work inside the PDF right now. Sorry about that. Okay, so let's go back to fertilization. All right, so with the whole process of fertilization, the kind of the first step that we're going to see after that hyperactivity is this docking station. And so keep in mind that when the heparin binding proteins were attached to the sperm head, shown here in the darker purple, and that it actually, when they were ripped off, they exposed the zona pellucida docking stations. And so the zona pellucida is going to be this area around the outside, of the oviduct, or not the oviduct, the oocyte. So it's kind of the cell membrane of the oocyte, if you will. And so the next layer in is going to be this paraviatine space. And then we've got some cytoplasm in here like a normal cell. And then kind of in the center, we've got the nucleus that is housing the oocyte, or the female contribution of the DNA. And so once the sperm cell has landed on this docking station, what it's actually doing is it is lining up these receptors so that when the zona pellucida 1, 2, and 3 docking stations are activated by the proteins on the outside of the sperm cell that actually will release acrosomal contents into the sperm cell. And that leads us to this acrosomal reaction. And so the acrosomal reaction, again, is a sub process within the bigger fertilization process. And this reaction is initiated by the zona pellucida 3 proteins binding and activation of that acrosome. And so the membrane will actually fuse and form vesicles, and this process is called vesiculation. So if you guys remember back to basic like cell anatomy or cell biology, vesicles are a big part of the cell. So there's lots of vesicles all the way through the cell. And so this is one instance where those vesicles become really, really important, is in this fertilization process. And so once vesiculation takes place, the contents of the acrosome are going to be released. And so those contents are enzymes, and enzymes are going to be activated to digest or cut away, eat away the zona pellucida of the oocyte. And so this is really similar to like heating up a really hot marble and putting it into a tin of butter. So it kind of just seeps through the acrosome or through the zona pellucida. And so this is what the acrosomal reaction looks like on the sperm cell. So this is before the acrosome occurs. This is during the acrosome reaction. So when binding takes place to the zona pellucida, and then we get all of those proteins that are just dissolving into that acrosomal cap, and our fusion proteins are going to be stripped away here. And so the next mini process after the acrosome reaction is the cortical reaction. And so this reaction, again, is another sub-process within the bigger process of fertilization. This is a heavily, heavily regulated process because we don't want fertilization to just take place willy-nilly, right? So if we get unregulated fertilization taking place, then the uterus does not have the ability to sustain all of those embryos. And so the cortical reaction, again, is another process where that is going to occur because of those proteins on the acrosomal head fusing together. And so the cortical granules are going to contain a protein or an enzyme called a protease. And those protease, in addition to peroxidases, are going to be released into the perivitine space. So it's like going in layer by layer into that oocyte. So we've been through the zona pellucida. Now these proteases and peroxidases are going to go into the perivitine space. And so at this point in time, a zona block is put in place on the oocyte, which means that no other sperm cell can fertilize that particular oocyte. So it blocks it from happening. And so this action blocks polyspermy from taking place. The Vittine's block then helps prevent other sperm cells from binding. And so what this looks like here is, again, we've got the zona pellucida up here towards the top. And so that's kind of layer number one. The sperm cell has already dissolved through the zona pellucida. It's now in this perivitine space. And here we actually are seeing that that inner acrosomal membrane occurring, plus the postnuclear cap, and we're starting to see all of these enzymes being released during that acrosomal reaction. And so the releasing of those enzymes will stimulate fusion proteins, which are going to bind and then further engulf the sperm cell. And once that has occurred, then we see fusion between the sperm and the egg membranes. Okay, so here's the sperm cells. So we know that the DNA is going to be in there. And then it will actually bind with a oocyte plasma membrane, and it's just being engulfed during this point in time. And also after the membrane fusion has taken place, we see a lot of condensing of the chromosomes or the DNA inside the sperm nucleus. And that is key for finishing up this fertilization process. And so that where we see fusion of the male and female pronuclei together is called syngamy. So that's a fun word to say. You can throw that one out at your next Jeopardy tournament. So syngamy is where the sperm and the nucleus are going to enter, or the sperm nucleus that has all the DNA from the male side is going to enter the egg, the cytoplasm of the egg, as a pronuclei. So one copy, haploid copy of DNA. So the DNA is going to condense so that those particular chromosomes can line up with the female complementary chromosome. And those two, that action of lining up and coming together is the pronuclei fusion. So we have male nuclei joining with female nuclei, male chromosome 1 finding female chromosome number 1 and fusing together. And that is the process of syngamy. And that finishes up the process of fertilization. So it is, again, I know that was a lot of information to take in a pretty short amount of time, and we're going to switch gears and we're going to talk about what happens after fertilization to that embryo. But the end point of fertilization is the development of a zygote. Okay, so a zygote is going to be a fertilized embryo. And so, it's going to be a fertilized oocyte. So any quick questions about fertilization really quickly that I can help you with? All right. It is a little overwhelming. That's why we're doing this just an abbreviated version to give you guys kind of a bird's eye view of it. But the big take home message here is that fertilization is highly, highly regulated. So it has processes inside of processes because we don't want that to be a process that just runs rampant. All right, so let's switch gears and talk about chapter 13 really briefly. And this is where we're going to talk about early embryogenesis. And just a second, let me get my screens figured out. Early embryogenesis and maternal recognition of pregnancy. And I'm going to just briefly go through early embryogenesis because I really want to get to maternal recognition of pregnancy because it's something that we have been talking about for the last few weeks. So, okay, come on. Where am I at? There we go. Can somebody just tell me, are you guys looking at a slideshow that says early embryogenesis, just the slide, not a note slide?
We see the note slide.
Okay, that's what I was afraid of. You would think that I would be able to run Zoom at this point in time, but I cannot. Okay, what about now?
It looks good now.
Okay, thank you. Appreciate the feedback. All right, so with early embryogenesis, keep in mind that the process of fertilization just took place, and our ending process of fertilization was the development of a zygote. And so what chapter 13 is really going to cover is the stages of embryonic development. And as I alluded to in the beginning of today's lecture, this is a process that you guys are somewhat familiar with the terminology because we talked about it really in depth in chapter 4. So when it comes to an embryo being able to attach, that's really the climax event for that embryo is it has to attach to the uterus so that it can develop placentomes, a shared blood supply, a place for nutrients to cross, and a place to grow. So the gestating process can actually happen. And so in order for that to happen, 4 major things have to take place. First and foremost is that the embryo has to develop within the confines of the zona pellucida. Okay, then we have to see hatching occur. So that's where the cells of the zygote replicate so many times that they can no longer be contained within the confines of the zona pellucida. So it hatches outside of the zona pellucida. In addition to that, we have to make sure that the female's endocrine system recognizes that there is a pregnancy present. Okay, and then finally, we have to see formation of the extra embryonic membrane. So this is like the formation of the placenta and the cotyledons. All right, so what this process looks like is once we see those pronuclei fuse through the process of syngamy, we end up with a zygote here. And so the zygote is where we see the 2 DNA sources, male and female, start to replicate in cells. And so this is the process of cleavage. So where the cells are going through mitotic, MIT, mitotic division. So like a copying machine, right? It just makes more copies, more copies, more copies of the same thing. And those daughter cells are then going to be termed blastomeres. And so that initial zygote fusion will go through one stage of mitosis and we'll get a 2-celled embryo. And then those 2 cells will go through mitosis again, and we will get a 4-celled embryo and then an 8-celled embryo. And once it gets to the 8-cell side here, we call that a morula. So the cell terminology is changing very rapidly here. And so that morula is then going to continue to go through mitosis and up until the 16-cell stage point. And all of these cells prior to being at a total of 16 cells are what we call totipotent. And that means that a single blastomere can actually give rise to a complete, fully formed individual. All right, so the cells don't have assignments yet. So the cells after the 16-cell stage become differentiated, meaning that they get directions of their job description. So they're going to be a liver cell or a skin cell or an embryonic cell or an amniotic cell, those type of things. So up until the 16-cell stage, they can be assigned to any type of tissue at that point in time. And so after this point in time, we see cells accumulate into an early blastocyst. And then we see the cells become so numerous that they hatch out of the confines of the zona pellucida here. And so that blastule is then going to start to just continue to develop over and over and over again through the process of mitosis. And so here's where we start seeing some similar terminology to chapter 4. So we have an intercellular mass here, that we have a blastosole here in the center, that is kind of the inner cavity. And then we have the trophoblast, which is going to be the cell structures along the outside here. And so once blasting occurs, Oh, I forgot to put that animation in. That's accurate. So the hatching is going to then lead into a hatched blastocele. And so the zona pellucida is going to completely go away at that point in time. And so just that's kind of an overview of what is happening. And so just a really quick comment about cell-to-cell communication here. In the morula and in the blastocyst, we actually see gap junctions and tight junctions forming. And so you guys may have heard of these things like in a biology class, but this is why they're important from a reproductive standpoint. So these tight junctions are going to be where cells are actually adhesed to one another. So they're glued to one another. That becomes really, really important for their place inside that zona pellucida. And then the gap junctions here outlined in yellow are where cells communicate to one another. All right, so and in between those cells, we're going to see fluid accumulate and start activating the sodium potassium pumping system that the rest of our body for the rest of our lives is going to depend on. And so this process really happens along the oviduct and into the uterine horn. So fertilization is going to take place clear down here at the ampulla isthmus junction. And then we're going to see the zygote be formed a little further down the oocyte or down the oviduct. And we go to a 2-cell, 4-cell, 8-cell morula stage and an early blastocyst. So for those of you guys that are familiar with the embryo transfer technology that we use in cattle and sheep and sometimes hogs, the terminology becomes really, really important because we end up flushing embryos out of our donor cows in the early blastocyst or the hatch blastocyst stage. And so ideally, we actually want them in a morula stage. If they get to the hatch blast, then they are not going to have a really high success rate in our recipient cows. All right, so our extra embryonic membranes are things that we've talked about before. So we've got a yolk sac, a chorion, an amnion, and an allantois. And so those are gonna eventually morph into our little embryo that actually starts looking like a little, or like taking the form of a little mini livestock species. And so the one thing that I do want to just talk briefly about is this process of maternal recognition of pregnancy, because it is very, very endocrine based. And so if you recall back to when we were talking about the cell signaling mechanisms and hormones, we need, in order for that embryo to attach, it first has to notify the uterus that it is there. And if there is no embryo in place, like fertilization did not successfully take place, then the uterus will produce prostaglandin F2 alpha. However, if an embryo is alive and there, then there's two ways that the conceptus can show the uterus that it is there. First of all, it can produce an anti-luteolic substance that will block PGF2 alpha, or it can produce a luteotropic substance that will maintain a UCL. And these are very species dependent. And so you'll see more information about this in the full lecture. So the blocking of PGF2 alpha is pretty common in like bovine species and sheep. So the embryo is going to produce interferon tau. Interferon tau is going to block PGF2 alpha from being produced. And so we get an endocrine layer like this bottom graph down here. So P4 is still really high. The conceptus notifies the dam of its presence. Oxytocin receptors and prostaglandin F2 alpha stay very low and will not trigger luteolysis to take place. However, if that embryo is unable to communicate that it is there and viable, then we see oxytocin receptors increase in presence. And with the increase in oxytocin receptors, we will also see an increase in the pulsatile production of prostaglandin F2 alpha. And that pulsatile production of PGF2 alpha is what will trigger luteolysis to take place. So it will go to the uterus or go to the CL on the ovary, it will lyse it, kill it, and we'll start the whole cycle all over again. So this is how that maternal recognition of pregnancy can trigger PGF2 alpha or not. All right, so that is a crash course in fertilization, or in what sperm is doing in the female tract, and also what is happening after fertilization takes place. So with that, guys, I'm going to leave, even though we've got a couple of minutes left, I want to make sure to answer any questions you have about turning in the group project, which is due this Friday, May 6th by end of day, or if you have any questions about the lecture material or the class in general. I'm going to leave it open for the rest of our time together.
Hi, Casey.
Hi, Linz. How's it going?
Good. How are you?
Good, thanks.
I just had a question about a part of the group project.
Okay.
So me and Haley are getting towards the end, and we're just having trouble understanding what you would like for the appendix part.
Okay.
We're trying to follow the criteria in the rubric, but we're just kind of starting to be repetitive, and we didn't know, and like it's also just really short.
Yeah.
And so we just want to make sure we're doing what you would like for that.
Yeah, no problem. So the appendix is really for materials that you don't want to take the time to fully explain in the bigger part of the project. So for example, if you're going to use like an estrus synchronization protocol, and the reason that you want to use it is because it was found to have favorable results in peer-reviewed literature, the appendix would be a place where that peer-reviewed literature could go.
Okay. Gotcha.
So the appendix is a really good place for any extra resources, or any, it's kind of like an extended lit review, if that makes sense.
Yeah. Okay. And so then the synchronization, would there be anything else that would, like, you could think off the top of your head?
Yeah, like nutrition stuff. If you were going to use, a different, so MGA is a really good example in beef cattle, right? So if you're doing like one of the heifer protocols and you were using MGA, you would want to justify why you're going to feed MGA, and you can do that with peer-reviewed literature.
Okay, so kind of going on more of a tangent.
Yeah.
To like justify.
Yeah, not necessarily a tangent to justify, but just to have extra resources. Like, okay, the appendix is a good place for me to look at, to know that you did your research and the decisions that you made. Does that make sense?
Yes.
Cool.
Okay, sounds good. And then I think that's it.
Great.
All right.
Thank you.
Thank you.
Any other questions, guys? Would love to answer them.
I do have a quick question.
Sure, Daphne, go ahead.
When it's talking about things like conception rate, such as like maybe the goal being like 75%.
Mm-hmm.
I don't know if I did my math correct, but I was trying to figure it out for like use.
Yeah.
So like, say for example, there's 200, and if there's a 75% conception rate, then trying to find like between singles and twins, would it be like a good rough estimate to say that we could expect a lamb crop of about maybe a little bit over 200, because it won't be double, right?
It depends on the breeds that you're using. So if you're using like a highly fertile breed, like a Finn sheep or something like that, that typically has quads, then you can get over 200%. Yeah. But typically industry average for for sheep, because they're a twinning species, is
Mhm.
usually between like 150 and 170. And that's a weaning percentage. So it's
Gotcha.
really, yeah, you may want to shift your metrics, Daphne, from conception rate to either pregnancy rate, which can be detected with an ultrasound, or
Gotcha.
to lambing rate, which is like live lambs on the ground. Or the better one from an economic standpoint is the pounds of lamb weaned per ewe exposed.
Pounds of lamb weaned per ewe exposed.
Yep.
Gotcha. Okay, so steer away from the conception rate and probably go to a pregnancy rate, which can be checked by ultrasound, and we could cite in a source or something like that.
Yep.
Okay, I get it.
Awesome. Great question.
Thank you. Have a great day.
You too. Thanks.
Thank you.
All right, guys. Any other questions? Hey, Jason.
Hi, yeah, I had a quick question. It's actually for the 199 application that you just sent me the other day.
Okay.
I have completed the campus defense driver training, but I don't know when it expires. There was never like an expiration date after I completed it.
4 years on that one.
Oh, it's 4 years. Okay.
Yep.
Got it.
Yep.
So then I guess I'll just put then 4 years
Perfect.
when I took it.
Yeah, if you have the date that you took it, that's perfect.
Okay. Okay, yeah. So then that, and then the only other question was, you said send my schedule as well in the email. Does that mean my future schedule for next semester?
Yeah, fall schedule if you've
Okay.
registered. If you haven't
Yeah.
registered, don't worry about it.
Okay, yeah, I have registered. There's one class I'm waitlisted for. I don't know if I'm gonna get in, but
Okay. Yeah, just
so-
make a note of when that could be.
okay, got it. Yeah, okay, I think that's all the questions. And then is there for the interview, do I have to bring a resume for that, or
So you're gonna send one in the email, right? Because didn't we ask for the application plus a resume?
no, you just said the application
Okay.
and the schedule. That's why I was
Okay, I thought in the instructions for the application there was a resume, but yeah, why don't you go ahead and bring one?
okay.
That'd be great.
Instead of emailing, or you want me to do both?
Do both. Yep.
Okay, got it.
Yeah, and then we'll let you know. I've got a whole bunch of applications already in, so I'm gonna work with the beef unit crew this afternoon, and we'll figure out a schedule for the 13th.
Okay, sounds good. I mean, yeah, and for me, at least, if you remember, I'm good